This is the third module in the 5-module Research and Developmental Psychology lecture. And what we're going to do now is we know now how to form a hypothesis, pick a sample, and collect data. In this module, we're going to focus on the experimental method as seen in laboratory experiments and the correlational method, which is typically conducted in real-world settings like elementary schools and the workplace. So where we're going to begin is the experimental method. And the experiment in an experimental method is the relationship between two or more variables is investigated by deliberately producing a change in one variable in a situation and observing the effects of that change on other aspects of the situation. Now I apologize if this gets a little too textbooky. That's where I lifted the definitions from. But we'll go ahead and try to make it a little more concrete later on. When you do run an experiment, you have at least two variables, so we'll focus on the easiest type of experiment, and that's just two factors that we're investigating. The first thing that you need when you're conducting an experiment is an independent variable. Now this is a variable that's manipulated so that its causal effects can be assessed. In other words, we want to see how it impacts other things. Those other things being a dependent variable. Dependent variables have differing effects on the behavior caused by the independent variable. So the independent variable is the hypothesized cause, and the dependent variable is the effect. Which cause and effect relationships are studied. Now this is a very important thing to note because the experimental method is really the only way that we can get at cause and effect. The later things that we'll talk about in this module, the correlational model, you cannot make that same assumption of cause and effect. Okay, so let's go on and look into the critical features regarding the experiment. The first is experimental manipulation. We use experimental manipulation to detect relationships between variables, and it requires the responses of at least two groups to be compared with each other. In one group, you will typically see the treatment. This is the manipulation implemented by the experimenter. The experimental group will be any group receiving a treatment, whereas the control group would be a group that receives no treatment. For example, weight loss has been a real big issue lately, and there are various dieting plans out there. If we wanted to do experimental research on it, we would go ahead and have or manipulate what someone eats through the diet. And so we would take two groups of people, we would randomly assign them to either a control group where we would not do anything other than telling people how to eat properly, and then we would have something like the Atkins diet, which would be the other one, and they would be falling into the experimental group. And then we'd have them strictly follow the Atkins diet. We would then compare the two, the experimental group to the control group, to see if there's any difference. So the independent variable would be the type of diet a person has. The dependent variable would be the impact on either the experimental or control group. So let's go on. Hopefully that clarified things a little bit. In experimental research, random assignment is very important. Now when we talk about random assignment, this is just like a random sampling that we talked about in earlier modules. Okay, random assignment of participants to experimental conditions is a way of ensuring that the treatment groups are similar in all respects at the outset. Because if we randomize everything, then we shouldn't run into issues, such as if we didn't randomize, maybe we would have people who are overweight and can lose weight somewhat easily in one group and people who are very skinny in the other. That would go into biasing the overall impact of the results. And so random assignment is usually the standard way that people are assigned to groups. Experimental control in a true experiment is where all factors other than the independent variables are controlled or held constant so they can't contribute to differences among the treatment group. Now, in the example I was giving you, it would be very difficult to have experimental control because we would have to control how much exercise the people had, what is their metabolism rate, and so there are a lot of things that are very difficult to have a true experimental condition, especially when you're talking about real-life settings. The solution for some of that is what they call a quasi-experiment. Quasi meaning experiment-like study that evaluates the effects of different treatments but does not necessarily randomly assign individuals to treatment groups. The limitations of this approach are fairly obvious in that there are uncontrolled differences among groups that could influence the results. It's just very difficult in a quasi-experimental study to have very hard conclusions because there's other factors that could explain it. In a true experimental study, you would have eliminated all of that and can say truly, this causes this. Okay, so it's a very important part of psychological research, experimental research, but unfortunately we can't always use that, either due to the expense or the type of experimenting we're doing. You can only imagine if people go in ahead and volunteered for a dieting study and then they get stuck in seclusion and everything about them is controlled and limited, including how much exercise they can do. It'd be very hard to find some willing people to contribute to that study, not to mention very expensive. So what we rely on a lot of times is something called correlational methods and correlational research. Okay, the correlational research generally involves determining whether two or more variables are related in a systematic way. Now, these variables are usually scored or measured within a scale of -1 to +1. So if you have a +1 correlation, that pretty much means that anytime one variable goes up, the other one does. With a -1 correlation, that means every time one variable goes up, the other one goes down. Usually you do not see perfect +1s or -1s. Typically in the real world, you don't see much getting above 0.5. Only in rare cases do you see it higher. For instance, the intelligence of family members, in other words, parents' intelligence is correlated to child's intelligence at 0.8 when you become an adult. And so when you see 0.8, that is extremely high anytime we're talking about real-world variables. It is important when we talk about correlational research. Just because something's highly correlated does not mean that one thing causes the other. And this is something I can't emphasize enough. Correlation does not mean causation. So when we have a correlation and we think it goes one way, it could very easily go another. So the directions could be reversed. Or a third variable could be influencing both relationships. So whenever you hear something have a high correlation, please don't jump on board and say, why, certainly this must be the cause of it. A perfect example of this is that there's a very high positive correlation between foot size and achievement in children. In other words, every time a child's foot size grows, their achievement tends to also go up. Now, does this mean that you want to start stretching children's feet so that they can become better achievers? Of course not. The reality is that when children are getting older, their feet grow, and so does their academic skills. It has nothing to do with your foot and your achievement, but there is still a correlation. So a correlation is really just talking about the relationship between the two variables, not the cause. So hopefully that's been drilled into your head now, and I only beat the dead horse a little longer. Okay, here's a nice little graphic that gets into both positive correlations, zero correlations, and negative correlations. You can see on part A of that slide with a positive correlation that as the variable y goes up, so does variable x. Zero correlation shows that it doesn't really matter what variable y does to predict variable x. And a negative correlation shows the higher that y gets, the lower that variable x gets. Okay? All right. You see lots of correlational studies out there, and one of the most useful right now is something called a meta-analysis. When we talk about a meta-analysis, this results in multiple studies addressing the same questions that can be synthesized to produce overall conclusions. These are like mega studies. You take the populations from a variety of studies that have been published, say 10 to 15, and you're able to take variables that are the same in all of those different studies and group them together. When you group those together, you develop something called an effect size that looks at the differences between groups. If you have an effect size of 0.2, we consider that a small effect size, and so it's not something that probably needs to be really looked into too much because it's fairly insignificant. If it's above 0.5, we call that a medium effect size, and it's something worth noting when they write up a study. And then finally at 0.8, we would consider this a very large effect size, and this is something that people would publicize and say, look, this seems to be the trend over 100,000 people. Anytime you can get large amounts of people to make conclusions, of course, the better it is. Now, several of the studies in your assignments will have meta-analyses done. I am not going to ask you to look at effect sizes so much, other than do glance at some of the tables of the meta-analysis, and just in general try to remember 0.2 small effect size, 0.5 medium, and 0.8 or above a large effect size. And don't try to get too confused because it is a confusing statistical technique, but you need to be aware that meta-analysis are out there and are used fairly frequently in developmental research. So that's pretty much this module. We reviewed the experimental method, which is conducted in more structured laboratory settings, and this allows us to establish a cause and effect relationship. It is the only method that allows us to do this. The correlational method, which is usually conducted in real-world settings, can help determine how variables are related, but does not allow us to make statements of causation, such as foot size causing intelligence in children. The next module will focus on 3 different designs used by developmental researchers to measure change over time.